Assalamu alaikum and welcome back to my YouTube channel Studyholic. Let's begin with our today's lecture, that is nucleus. As we know that the nucleus is the most important part of a cell, so I'll be discussing it in detail, and it's very important for the for understanding all the organelles is one side, but the nucleus is the most important one. So let's start with our discussion. Okay, so talking about the location, firstly about the location. In animal cells, the nucleus is present in the center. But if we talk about the plant cells, in adult plant cells, the nucleus is present away from the center because of the presence of the large central vacuole in the plant cell, and that pushes the nucleus to the peripheries. Now let's have a look onto the parts of the nucleus. The eukaryotic nucleus basically consists of nuclear envelope, nucleoplasm, nucleoli, and chromatin. So let's have a detailed discussion about all of the parts. I have attached the diagram of the nucleus, which basically dictates all of the parts, which I'll be further discussing. Let's have a look onto the nuclear envelope, which I'll be starting first in my discussion. Okay, so I'm drawing a nucleus, a gross diagram. This is nucleus. Let's suppose that it's nucleus. So the blue one is the first membrane, and as we know that the nucleus is a double membrane covering, so the red one is the second covering. So the nuclear envelope is basically a double membrane covering, and that makes up the boundary of the nucleus. And both of the membranes, the nuclear envelope is basically made of the two membrane, that is its bilayer, and these both membranes are separated by a fluid filled perinuclear space. This blue one is this fluid between the two membranes, and this is known as perinuclear space. So this was about nuclear envelope, that it is bilayer. It is separated by the perinuclear space. Now let's talk about the composition of the membrane. The nuclear envelope is composed of lipid bilayer and proteins. And if we talk about the outer membrane of the nuclear envelope, it is basically covered with ribosomes, and thus the outer membrane is also connected with the membranes of the ER, that is endoplasmic reticulum. If you want to know the prior knowledge about the endoplasmic reticulum and the ribosomes, so I'll be attaching the video lecture I have discussed in detail in earlier lectures. You can watch them and then you can switch to this one so that you can get the clear concept. Okay, so jot down all the points. First of all, I talked that the nuclear envelope is bilayer. Then these two membranes are separated by a space, fluid-filled space, that is known as the perinuclear space. And then the nuclear envelope is also covered with ribosomes, and that thus it is connected with the membranes of endoplasmic reticulum. So let's talk more about the specification. There are also the nuclear envelope has also numerous pores, and hence as these are on the nucleus, so these pores are known as nuclear pores. And these pores are composed of nucleoporin. This is nucleoporin is a specialized transport protein that helps in the transportation. So we call this it as nucleoporin. You can you keep writing the points of all the specification of nuclear envelope. It will help you learn fast. Okay, so what is the job of the nuclear pore? Basically, nuclear pore is the point at which the both membranes are connected. Um, if I talk about this is pore, and this pore is connecting the two membranes, like this membrane, here is the pore, and it is making a link between the two membranes. So why this nucleopore link and the connection between the two membranes is required? Basically, this makes the nucleocytoplasmic exchange of materials. How? The inner one, this is the nucleus, and there is a this nucleopore basically helps the exchange of the materials between the nucleus and outside, that is cytoplasm. This one is cytoplasm, get it with the blue, and red one is the nucleus, consider it. Okay, what are the materials that are exchanged between the nucleus and cytoplasm? Okay, the first one, RNA and ribosomal proteins are moved from nucleus to the cytoplasm. Let me write over here. Okay, I've written over here. RNA and ribosomal proteins are transported from nucleus to the cytoplasm. And what are the materials that are moving into the nucleus? Those are These are the ones, the DNA polymerase, carbohydrates, lipids, which move from the cytoplasm into the nucleus. Okay. Okay, so that was all about the nuclear envelope. We have discussed some important points. First of all, the nuclear envelope is double membraned. It is separated by a filled space, that is perinuclear space. Then we talk about its composition, and lastly, we discuss about the nuclear pores, how they make a connection between the double membranes of the nuclear envelope, and what are the materials that are exchanged between the cytoplasm and nucleus. So let's move on to the next part, that is nucleoplasm. Let's move back to the diagram so you can have a look onto the real diagram, because my diagram I don't think so it's much appreciating. You can please let me know in the comment how it is. Okay, so this is nucleoplasm. Let's discuss it in detail. Okay, so nucleoplasm is basically transparent, semi fluid ground substance that is formed of a mixture of proteins, enzymes. Enzymes include DNA and RNA polymerase, free nucleotide, and some metal ions, that is magnesium, and that are responsible for the synthesis of the DNA and RNA. So, nucleoplasm is basically responsible for the synthesis of the DNA and RNAs. The nucleoplasm also contains histone and non-histone proteins that are involved in mitosis basically. So the nucleoplasm is similar to cytoplasm, but it is slightly different from the cytoplasm because it is involved in the formation of the DNA and RNAs. So that was all about it. Now let's move on to the nucleolus, the next one. This is nucleolus in yellow one. As you can clearly see over here that it is a non-membrane bounded structure and it is present in nucleoplasm. The number of the nucleolus varies a cell to cell, and there may be only one or there may be many. So when does the nucleolus basically appears? It appears during the interphase, and then it disappears during the cell division. So if we talk about its structure, the periphery of the nucleolus basically is granular area, and the central area is basically fibrillar area. The periphery is the granular area that contains ribosomal subunits. The central one is the fibrillar area that contains ribosomal RNAs and ribosomal DNAs. So this specification, due to this, the nucleolus is involved in the construction of ribosome. The nucleoplasm was involved in the formation of the DNAs and RNAs, and the nucleolus is basically involved in the construction of ribosomes. Learn it by heart, that what are the functions, what are the structures. These are gonna help you in lifetime. Okay, so let's move on to our last one, that is chromatin. Chromatin is basically a thin thread-like structure, and it is made up of the DNA and protein. Let's illustrate over. It is like a web kind of like this, and it is made of the DNA and proteins. And how these chromatin basically change into the chromosomes, and when does this takes place? During the cell division, this takes place that the chromatin fibers they condense and coil up into the separate structures, and these separate structures are known as chromosomes. Let me draw over here. This is the transformation. This is the chromosome in red one, and they are thick enough that they can be seen in the light microscope. So as you can see over here, that the chromosome, the typical chromosome basically contains the two strands. These are the two strands, and they are called as chromatids, and they are attached at each other. These are the two chromatids, and the point of the attachment is known as centromere. And the point of the attachment where the centromere is attached is known as primary construction. So the chromosome basically contains a two strand that is known as chromatids, and the point of attachment is called a centromere, and the where the centromere is attached, that area is known as primary construction. Centromere is basically involved in the movement of the chromosomes during cell division. It helps in their movement, and the centromeres basically consist of a complex of kinetochore proteins present on the opposite side of the construction. This is the construction, this the V-shaped, this one, right? And on the each side of the construction, there is a protein attached to the centromere, and this protein is known as kinetochore, right? What is the function of kinetochore? Basically, the kinetochore forms the site of the attachment for a single microtubule during the cell division. So this kinetochore basically functions during the cell division. As we know that first there was chromatin, and during the cell division the chromatin converted into chromosomes. The chromosome has two strands that is known as chromatid, and the point of the attachment of the two chromatids is known as centromere. The centromere is located in a construction that is known as the primary construction, and the centromere has kinetochores on either side of the construction, and this kinetochore helps in the attachment of single microtubule during the cell division. So that sums up all the structure. And if we talk about not only the true chromosomes have the single point of the attachment, though, that is known as the centromere primary construction, they may have another point of the union along the length, and that point, the other point other than the primary construction, is known as secondary construction or the nucleolar organizer. Let's suppose the central connection was known as the primary construction. If the chromatids are attached on the other side, that would be known as the secondary construction, or we would be naming it as the nucleolar organizer. And this construction, this point, basically gives rise to nucleoli during interphase. Okay, so that was all about the lecture today, and the nucleus was all about here. And now let's move on to the brainstorming session. Okay, moving on to the first one. Membranes of the nuclear envelope are separated by pericardium, perinucleolus, perinuclear, and periseptum. Okay, so let's just think about the answers. Surprisingly, is no one option that is right. It's perinuclear space. The E1, I put this just to have a brainstorming of yours, that have you really listened to it or not? Okay, let's move on to the next one. It is a network of thread like structures: chromosomes, centromere, chromatin, chromatids. This one's gonna be easy one. Yep, it's chromatin. So that was all about the today's lecture, and if you find it helpful, so don't forget to like, share my video, and subscribe to my channel. Thank you. Stay tuned for my next lectures.